2, 3. And good morning from the RV Falkor, where once again we have the pleasure in bringing to you one of our water dives of the ROV SuBastian on back of the RV Falkor. We're still in the Coral Sea Marine Park as we were yesterday in preceding days. My name is Nichol from Geoscience Australia, and today we are currently located on the eastern edge of Tregrosse Reef, which is some 480 kilometres due east of Townsville, northeast Queensland, Australia. The SuBastian is now descending through 120 metres water depth almost. Our target depth on today's dive will start at around 150 metres depth and run up towards 150 through the day. Before we get to the seabed, we'll have the time allocated to what we call a mid-water dive, where we take our time to descend through the water column, in particular to observe and potentially sample water plankton with a focus on the jellyfish, the larger forms of the plankton that we see in the column. And once again today we have the pleasure of welcoming Dr. Dougal Lindsay from Japan Agency of Marine Science and Technology in Japan, who's sitting in his home in Yokohama, south of Tokyo in Japan.
Wow.
He's already seeing stuff come online.
The Siphonophore. The float and nectarosome is bottom right of the animal there. And the cyphosome, or the feeding apparatus with all those long tentacles coming off, is coming up from there.
Good morning, Dougal. How are you?
G'day, Scott. Konnichiwa.
Konnichiwa. Lovely.
Nice to meet you. Howdy. Very international team.
We should. That's right.
Because of coronavirus-related travel restrictions, rather than actually being there on the ship, I am here in Japan and watching the video being streamed live and giving narration and instructing on which animals to be sampled, etc., which is amazing. We can do this now. I work for the Japan Agency of Marine Science and Technology, JAMSTEC, mostly working on jellyfish, gelatinous animals. Jellyfish or jellies actually encompasses a large range of different taxa in different phyla even. So anything that's squishy, basically. And we see a lot of those squishy animals when we're out doing mid-water dives, because especially in areas where there's not very much upwelling or there's not very much phytoplankton in the surface layers, the gelatinous animals pretty much take over, because they need a small amount of organic matter in their bodies. Their bodies can be mostly water, and they can still reach fairly large sizes, allowing them to predate on a much larger volume in the water column. So we are probably going to see a lot of filter feeders, which have evolved to eat the very small particles floating around in the water. Many of these are gelatinous, although it also includes things like the pteropod snails, the sea angels, or the sea butterflies is another name for them, which make feeding webs. But it also includes the tunicates, the jellies with stinging cells, the nidarians, and the jellies with coloblasts, or things that are like fly paper on their tentacles, allowing them to stick their prey to catch it. So we should be seeing all of those on our way down. Manipulate arm is presently, I think, trying to get hold of the calibration board, which is a little black and white checkerboard-like board, which we're using to calibrate a pair of stereo pair of cameras, which we have on the SuBastian. The SuBastian has quite a few different cameras on board. The camera you're seeing streamed to you now is the main 4K camera. With this, we can zoom in on small animals, and we can pan and tilt, which means move left and right, or tilt is up and down, so that we can follow the animals in three-dimensional space, trying to keep them in focus and getting information on their morphology. As well as this main camera, we have situational cameras, such as there's one that's pointing down, which the pilot is probably using right now to see if they can see where to grab the handle for the calibration board. And there's other live streaming cameras such as this to provide situational awareness. And we also have a stereo GoPro system on board. And this has a right and a left camera, and we can map in three-dimensional space every pixel in the left-hand camera and which pixel it corresponds to on the right-hand camera. And by doing this in special software later on, when we click on the same point, say the head of a fish and the tail of a fish, in both of those camera feeds, we can then tell how large the fish was. We can also tell things like the distance between various planktonic organisms. So if we see a large school of krill, for example, we can click on each krill individually and say, the distance between them is 33 centimeters. Therefore, there's this many of them in a cubic meter of water. And this allows us to get quantitative data on the midwater animals. So we have the calibration board out now, and it will be moved, tilted, and moved around in the field of view of the cameras to try and get data that will allow us to map out each pixel in each camera. Because the GoPro cameras do not have a live video feed, this is a little bit of trial and error, hoping that the board is in front of the cameras as we're moving it. They have a very wide field of view, which is one reason why these are the cameras we've chosen, because while we're zoomed in or focused on a particular animal using the main 4K camera, we still want to see what other animals are occurring around it. Because the environment that is important for the animals, they might like certain depths or temperatures or salinities or oxygen levels, but they also are influenced by the abundances and types of their prey and of their predators. So as well as getting information on the occurrence of the animals themselves, we also would like to know what the prey field is like quantitatively to infer how important that is to the observed distributions of the animals. Okay. We have a variety of different angles there. We'll put that board back in the sample basket. What's that? Oh, there's it. Still here at 110 meters, keeping the same depth so we don't miss any data points while we're doing this calibration. With fancy manipulator work going on there. The sample basket, as you can see there, is being retracted. There's a hydraulic piston system set up there so that we can move it into and out of the field of view. This way, when we're doing midwater ops, like right now, we can pull it in underneath the ROV and give ourselves an unobstructed view of the water in front of us. And when we need to sample something, we can push it back out again and make it more accessible to the arms, and also so we can actually see what's going in each of the boxes. That's a really neat feature to have that, so we don't spend all of our dive looking at core sampler handles and boxes, which is often the case in ROV dives. We're doing a long, slow, oblique descent here, trying to get the marine snow particles, which are those little white dots you see flowing through the video stream, trying to get these marine snow particles so that they collide with the camera, just like bugs collide with your windshield. This is so as we go down through the water, we'll get at least one frame where each particle is in focus, hopefully allowing us to ID what the animals are, assuming they're large enough. There's enough pixels there that will help us identify them. The range of different particles in the water, it looks like most of them are fairly small so far. But from time to time at the stem, you can see strings of mucus or larger marine snow particles as well. The marine snow is produced by looks like a copepod there. So one of the challenges, Siphonophore. This is the cyclosome. To the bottom left there, that looked to be like swimming bells. It looked to be a Calibration Siphonophore. So that's a group of Siphonophores that has swimming bells but no float. So they can swim around, but they can't change their buoyancy by increasing or decreasing the amount of carbon monoxide gas inside their float, which will allow them to move up and down in the water column. Normally, the Siphonophores that vertically migrate are those that can change the amount of gas in their float. So just by changing the amount of gas, they can float up to the surface without actually having to use any energy, energy for swimming to get there. And then they just bubble the carbon monoxide out of the top of the float or the bottom of the float, depending on the taxa, so that they can sink back down to depth again. The Calibration Siphonophores, like the one we just saw, only have swimming bells, so they can't do that. The best they can do is change the concentrations of various ions within the jelly of their bells. If they increase the concentration of light ions, which are lighter or less dense than the ions in the seawater around them, they can become positively buoyant and float up shallower, and vice versa for deep. There's a marine snow particle, looks like the discarded feeding filter of an oikoplurid larvation, which is a little tunicate, like a sea squirt, except it only develops to what would be considered the larval form in regular benthic sea squirts. So it looks like a little gelatinous tadpole. And they produce feeding filters, which they sit inside this little house. Oh, there's a Siphonophore. And filter the marine snow from the water in order to feed. We should be seeing more of them as we dive today.
Dougal, it's Rob here. Just to let you know, in the next half hour, we'll be sharing a Zoom feed for some outreach back to the United States with the students we have on board. So we're sharing bandwidth here. We'll be quiet mostly here in the control room while that's happening. Feel free to talk from your end, of course.
Roger that. Would they be university students or high school?
Yeah, I think it's a mix. Yeah, I think it's a mix from the USA mostly, but we're zooming in with Jeremy, Gemma, and Antonella from on board.
Roger that. Which is another really great thing you can do when you have this video streaming capability for ocean surveys. It's even possible if we see an interesting squid, for example, and we can stay on it for long enough, we can pop off a message or phone an expert in the Smithsonian or some other institute and say, hey, can you look at the feed we have here? We think this is new. Should we sample it? What should we be videotaping in order to tell what species it is? Another Calicophora and Siphonophore here. 15 or 20 stomachs, each with its own tentacle. And the swimming bells, see if we can make out whether they are rounded or pointed like a triangular prism. Will give us a good idea which family it belongs to. Looks like the nectaphores are right hand, top right there. It's looking like a Prayed, Calicophora and Siphonophore belonging to the family Praeidae. If we're able to get a good in focus shot somewhere in there, we'll be looking for the somatocysts inside the nectaphores, as well as the general shape of them. And if the somatocysts embedded in the jelly of the bell have two large spherical bulbs on them, then it would be Desmophysinectins, which we have seen a few of during the present surveys at depths such as this, which would probably be my guess. So we'll see with the high quality video that we get later on, as well as this video we're streaming here, which has to necessarily be quite compressed and light in order to send it overseas or anywhere really, so we can all look at it real time. We're also recording the video at high quality in a different codec, which is far less compressed. Here's another one. This is also a Calicophora and Siphonophore. Looks like the same species we just saw. And we're focusing in on the nectaphores here. To see what shape they are. Yeah. Sometimes the nectaphores, the somatocysts inside can be branched in a regular, like fern-like pattern of branching. Sometimes they're not embedded in the jelly, but rather just lie pretty much flush to the outside of the bell, where the tentacles, the cytosome is attached. And these are the characters we use to tell the genus. Looks like you're probably getting good video there. And so by looking at the, we use a ProRes codec to record gelatinous animals during our midwater dives. Some codecs will compress the black end of the spectrum and leave the skin tones relatively intact. But we need to use a codec that allows us to see these mostly transparent animals that are almost the same color as their background. To be able to make out their morphology, we need to be able to see uncompressed in the blacks and the blues of the spectrum, which is why we're using the ProRes codec instead of one of the more common internet streaming kinds of codecs for recording the video. Okay, we're at 133 meters now, still moving through the water column, trying to get an idea of what animals are living here. This depth is still fairly brightly lit if you're a midwater organism. I believe, Rob, that there's still photosynthetic organisms around on the benthos at this depth, perhaps?
Yeah, that's right. Yeah, I mean, the waters are clear here. We've got the red algae, the lithothamnian crustose algae, photosynthesizes. We've detected that anywhere around between 200 and 220 meters depth. So that's around about 0.5% of available photosynthesis, light available for photosynthesis, but that's kind of the lower limit here. It's still very deep though compared to any other parts of the world. Just shows how clear these waters are.
So yes, if there's enough light for photosynthesis, there's definitely enough light for animals that have evolved to live in the murky twilight of the mid-water to be able to see their prey. Obviously, they'll see the ROV because we have our lights on so that we can see things. But the normal distributional depth of animals is often determined to a large degree by the amount of light. Because there are no rocks to hide under, no seaweed or wood to hide behind, you're basically out there floating in the blue, visible to everything that wants to come and eat you. So the animals are usually very small, so they don't, they're not very visible. They don't show up. If you're big, you're more easily seen. They're also usually fairly transparent because, again, to make them harder to see in the murky twilight. So very small, transparent animals are what there is most of here at these depths. And they're very difficult to find as we drive our several ton heavy hunk of steel with thrusters on, carrying a great big umbilical cable which is joined to the ship, and we're trying to find these little sub-centimeter or like centimeter long animals and stop on them in time that we can videotape them and record what they are. So it's a very challenging bit of survey work we're doing here. As we get deeper, the animals will tend to become larger because it's they don't need to hide or be small, as small anymore. And as we get deeper, there's less and less food. So the animals have to be larger so that when they go through periods without a meal, they can absorb their body mass and stay alive. Ooh, Physonex Siphonophore. Look like there. And another. Many stomachs. Make sure we can see the float. See, it was a Physonex. Coming up the middle of the screen there. Another Siphonophore, it looks like. Still in feeding position. And pulling in its tentacles now. Does that still have a nectosome attached? See any swimming gulls?
Doesn't look like it. Doesn't look like it there, Dougal, but at the very back of it thickens out though. Might be the nectophores.
Oh, there it is. Yeah. So it's a Physonex. Oh, just reversed. That bright white spot in the top left of the gelatinous. There we go. That gelatinous, slightly thick finger-like thing there. That's a biserially arranged pairs of swimming gulls. And at the left point, you can see the bright white reflection from the float. Looks like there may be some parasitic amphipods or something attached between the nectophores there, the swimming gulls. And then the tail or cyphosome is coming out the top right of that slightly curved finger-like nectosome. And the repeating chlamydial units each with their own stomach and tentacle and their own reproductive organs, protective organs, etc., are all splayed out in feeding position. Physonect Siphonophore. Hopefully when we look at the high resolution uncompressed video later, we'll be able to see the structure of the nectaphores, the swimming bells, and tell what group of Physonect Siphonophores that belong to. It is very hard to tell the species of Siphonophore from in situ video because most of the descriptions occurring in the literature when the species was first described are based on one or two pieces that were caught in a plankton net. And there's no description at all of the macromorphology of the colony itself, which makes it very difficult to just look at the colony and say, that's this animal. So in the years that we've been doing ROV or manned submersible dives, we've been collecting videos such as this and specimens to tell what colony macromorphology equates to which species, to be able to do surveys using video. Here's another Physonect Siphonophore. Quite a few of them around. This looks like it could be no. The jury's still out. If we can image the nectaphore side on, it's often possible to tell whether the nectar sac inside is a T-shaped or Y-shaped or just fills the nectaphore entirely, which will give us an idea of which family of Physonect Siphonophores it belongs to. I can definitely tell what it isn't. It's not a Forscalia, for example. Oh, that's nice. It's an Agalmatidae sensulato. Is there any chance we can suck one of these up, or are we too close to them? There's quite a few of them around, isn't there?
Yeah, we're going to try that, Dougal.
Thanks. So these animals are basically a colony of polyps, and the adult jelly, or the adult medusa, medusoid stage, is permanently attached to that polyp colony down on the tail, or the cytosome, which has all those stomachs and tentacles on it. In the Calibration Siphonophores, the adult stage can become released from the tail and swim around by itself. This is called the eudoxid stage of a Calibration Siphonophore, and it's the sexual stage, which produces the new larvae that develop into the asexual stage, which is the nice long polyp colony. The sexual stage only has one stomach and one tentacle, whereas the asexual stage is called the polygastric stage because it has many stomachs. Depending on the exact group of Siphonophores, these can be tens of meters in length. When one catches a Siphonophore colony using a plankton net, all the pieces are broken up and it becomes a jigsaw puzzle, which you have to put together again later. And most times you catch more than one individual in the same net, and multiple species as well. So you're never sure which part goes with what. The beauty of ROV collections such as this is we can collect an entire colony And we are sure that every single zooid that's in that canister all belong to the same colony, which is great for doing taxonomic work, redescribing the species or describing new species. Oh, he's pulled in all the parts that will cause drag, and he's hightailing it out, trying to escape from us.
Like chasing a ramen noodle.
Yes. Ramen doesn't escape from you. There he is. Looks to be quite a small colony and quite robust. Even with all that thruster watch, he's still in one piece. So we might guess that they can occur up near the surface, maybe at night. They've got a float that would allow them to vertically migrate. They don't fall apart suddenly, suggesting they are okay with wave action or with fish swimming past them at high speeds. Total conjecture, of course. I think this would make a great video game. It's going to be challenging to, looking at the video later, to make sure this is the same animal that we've got all the video of. It seems to be. He's in front of us. Yeah. Just gone inside the light shadow. The entire colony must be something like 10 centimeters in length, quite small. Oh, did we catch him?
Got him.
Excellent.
We got Ben and Cody on ROV Islets this morning, assisted by Nelson on the audio desk.
Great work. Those little guys are very hard to catch. Right, I can make my screen. There we go. Are we zoomed completely out right now? Yep, looks like that. Oh, there's a nice light. So in addition to the main pan-tiltable, zoomable camera that we're streaming right now, we have a stereo GoPro camera system, and we also have a couple of regular single GoPro cameras pointing at different angles. So one of them's pointing down. This is when we get steep floor that is not a vertical cliff. We can get a video of the seafloor looking directly down on it. Also, when we're doing a descent, it's often good to find jellyfish as well. That's a Siphonophore colony again, look like another Physonet. There's quite a lot of them around. I think that's the most Siphonophores we've seen at such shallow depths during the present survey series. There's another one.
Dougal, just let us know if you want us to try and slip one up.
Roger that. There's so many of them here, you may have them showing up in your multi-beam data as noise. With their acoustically reflective floats.
Yeah, that's right. We have actually seen layers like that at night time. Yeah, Nelson from the in the multi-beam team was saying last night it's quite noisy in the data.
Yeah, so many of those. There's another one. And another. Wow. And another. Kind of hard to get a handle on if they're all the same species or not, though. So forward in the water.
So that leads to a question I had, Dougal, in terms of the general location we're in this morning. In comparison to the other dives I've done with you, where, say, we were several hundred kilometres to the west in the submarine canyons and diving in much deeper water down to 1800, and we had that influence of the Antarctic intermediate waters coming into play. How are those sites compared, do you think, to where we are today, where it's much shallower and much further out offshore?
Well, I guess we'll find out during the course of our survey. But the Cairns Seamounts, I remember, had many filter feeders, Tunicates. Once we get good video of this, it'd be great if we could throw them off as well.
So we'll sample this one.
Yeah. So yeah, there are lots of Tunicates, which makes me think the waters are very productive. Oh, we left the feed here.
Just gone dark a little bit, Dougal. Just adjusting the lights, I expect.
Here, all these continents, Siphonophores, I'd assume there's lots of small crustaceans around that they are able to feed on, which means there's a lot of, rather than very small phytoplankton in the water. Maybe
there we go.
Something else.
That's better. So I would like to sample this fella.
Yeah, that would be great. Oh, there's heaps of them around. Oh, I wonder if it's a Ninomia species.
So we're about to set up for sampling, Dougal.
Okay.
There's 4 of them, Cody says. Take. So that gives a.
This is very thin. They all look to be the same. Very thin cyphosome. The bracts don't really stand out in the video, so they could be. It could be an anomia. They tend to have very small bracts. Currently in the Pacific, there's only one species of Ninomia, Ninomia bayuga, which is officially recognized. But this is based on fairly mashed up samples caught in nets. So see if we can collect one here and get some material for DNA barcoding to see if the sequence matches up and see if there's any morphological differences between it and some other species. Putative species that are now referred to by Ninomia bayuga, the scientific name. We have no samples of this species from off Australia, so this will be great if we are successful in capturing. It's certainly very active, aren't they? Oh yeah. I think that went in, didn't it?
Yeah, we landed that.
Awesome. So that's the whole colony. You can get all the pieces and know they all belong to that one colony.
Fantastic. Confirming Jar in a moment.
Okay. So 4 on the screen at a time, that's very high population density. Hopefully the stereo images will allow.
Do we have it in 2? 2 is confirmed.
Awesome. Yep.
That's a pretty good hit rate so far this morning for you, Dougal.
Yeah.
You want to go and give us control? I have to do it.
And one of our other target species, Solmissus, the dinner plate jelly, is known to eat that Siphonophore species. So the predators, the larger jellies that'll be eating that Siphonophore should be around as well. We have found, though, that these Physonect Siphonophores can be important prey for a range of different predators. When you open up the stomach contents of animals, fish, for example, the jellies are digested very rapidly because they're mostly made of water and they don't have any hard bits like scales or shrimp exoskeletons, for example, which remain in the stomach after a lot of digestion has taken place. There's another one, Ninomia. Well, Physonect Siphonophore, we'll call them for now. We'll be able to tell what they are when we look at the specimen later. There's another. It's phenomenal, the number of these animals. We have 2 specimens now, so we can just keep on driving past them and just like counting them as we go.
Roger that, thank you.
So if you see some that do not have the long nectasome, the long biserially arranged swimming bells, maybe a large Calicopheren, a Prayid or something, we can stop on that. Very large 2 opposed swimming bells. They could be possible predators of the Ninomia. And we still need to identify what species of those there are here. But hopefully our stereo cameras will allow us to compute how many of these Ninomia occur in a cubic meter. Possibly the sonar give a hand with that oikofluid larvation. It's another Physonect, putative Ninomia. Man, I don't think we've seen any of these Ninomia at any of the other stations. Oh, this is different. That's an Agalma okini, I think. Certainly looks like one. See if we can get a little bit closer. See whether the cytosome at a distance looks white. As we get closer, we should be able to see whether it's orange, reddish tinted or not. The red gets absorbed fairly rapidly by the salt water. Closer we get, the more red there will be showing up in the animals. A very thick cyphosome on this, and the very compact form. It's even shorter than the nectosome, the swimming gull containing area of the colony. Certainly narrows down what it might be. Nice.
Dougal, is this a Siphonophore? It has that nectosome look about it. So just explain again what this is.
So this is a Galma okini. It's a Physonex Siphonophore. The left part of the animal there is a series of pairs of swimming gulls opposed to each other. And the leftmost zooid, or the topmost zooid on the colony, is the float. It has the float is actually a zooid which has a very, very long tube attached to it. And this tube is kind of like the artery, the main artery of the animal, which it uses to share nutrients and energy around the colony. And the compacted tail on that is the cyphosome. It's very different. There's an oikopleurid. It's very different to this one that just went through the bottom of the screen there, which could possibly be Nenomia, which has very widely spaced stomachs and very small transparent bracts, which are protecting the stomachs and the reproductive organs from being eaten. The Agalma okini we just saw, the bracts are very well developed and they interlock in a Jenga kind of a fashion. It's another putative Nenomia. Yeah. That's awesome. So the Agalma had interlocking Jenga-like protective bracts, which cover the gastrozoids, the stomachs, and the gonozoids, the reproductive organs. Yeah, is that a cell? To stop them being eaten by fish or shrimp or things that see a full stomach and say, "Ooh, I think I'll eat that stomach complete with whatever's inside it as an easy meal." The Nenomia colonies look to have all their tentacles out. They're feeding, so they don't only feed during the night in surface waters. It looks like they're feeding down here during the day as well. And because the tentacles are all splayed out in a three-dimensional structure that hopefully we'll also be able to... Oh, what do we have here? That looks more like a Calicophoran cyphosome, but it looks to be missing the swimming gulls, the nectosome. Yeah, that's a prayed tail. The cyphosome can actually survive for quite a long time without the swimming gulls. That's cool. Because they can ingest prey and they can reproduce and protect themselves from being eaten using the bracts. Oh, wow. Can you see any pulsing or opening and closing of the zooids there? Some Calicophorans have special swimming bells on the cyphosome, which will pulse and propel the animal. Others, they will just be large, rounded bracts that are protecting the stomachs. I wonder what this is. Hopefully we'll be able to tell that in the high-quality video later. So there's a very great diversity of siphonophores living in the midwater, and a large diversity in their reproductive tactics as well. Now, some of the colonies are hermaphroditic, which means they have male and female sexual units on them. Others, the colony is totally male or totally female. They have separate sexes. how these colonies with separate sexes actually meet each other in the three-dimensional vast space of the mid-water and reproduce is still a mystery. It's like another Ninomia there. A lot of marine snow particles we're seeing here. And one would assume with the large number of predators, that was a Chitingnathe or arrowworm. Many of these particles are probably living animals such as copepods or ostracods, another Ninomia. As the ROV gets close, you can see some of the particles suddenly jumping in the water. Ooh, what's that? Medusa, perhaps. Another Ninomia. Liquid fluid, lavation house. There's a little crustacean there. Ninomia. Wow. So different to any of the other stations we've been surveying so far this cruise. I think we can safely say that you can't just pick an oceanic area on the Great Barrier Reef and assume that the midwater community is the same no matter where you dive. Very large differences in community composition between dives here. That's another Ninomia. It's an interesting posture. Looks like it's actively sinking. Must have taken all the gas out of its float and it's heading down, trailing its cyphosome behind it. That bright white dot on the bottom there is its pneumatophore or float. Another one. None of the stomachs are shrimp-shaped or krill-shaped. So if any of them have eaten anything, they look to be small copepods or ostracods. Mostly it looks like the stomachs are not full, but in order to have a population size such as this, there must be quite a lot of food around. Holy mackerel, look at them all. Just amazing.
Dougal, just to give us a sense of scale here, when they're fully expanded like this, what's the sort of size we're talking about?
Not above the Physinect in the top of the screen, it looked like there was a Medusa. Possibly a Narca Medusa. Go up just a little bit more. The entire colony is probably only, well, depending on whether it's contracted when it's escaping, it might be something like 10 centimeters long. Could be 15 or 20 when it's fully expanded. So these are quite small, fast-moving, fairly, what's that? It's an liquid fluid house, a larvation. Quite small Physinect colonies as far as Physinects go. This is interesting. Is this a Collidarian colony, perhaps? Going a bit closer, we'll see if it's a massive jelly with points scattered all through it or not. It's got no repetitive repeating structure, so I'd say it's not the ghost tail of a Siphonophore. it seems to be too, I don't think it's a mucus strand because it's staying together like that. There's a single-celled animal called a collidarian that produce large gelatinous colonies. And that may be what we're looking at here. They're normally a lot shallower than this. We're down at 226 meters. Normally, scuba divers or snorkelers would be able to see such colonies. And they often have copepods or amphipods attached to them, resting, so they don't have to stay swimming and floating all the time. I think we'll have a frame or two in focus there. Okay, I think that was a collidarian. So many phosphinates. When they occur in large numbers such as this, you can really imagine how the Navy back when they were doing surveys of the seafloor or looking for submarines hiding around in the mid-water or seeing these false bottoms. Can you really imagine how that might happen?
Yeah, that's right. And these vertically migrate, so they would actually see in their echo sounders the seafloor rising as the sun was setting, and they hit the brakes thinking they're about to hit some shoal, but it was just this deep scattering layer vertically migrating.
Was it a patch? Have we come out the other side of it, I wonder? Is this another collidarian colony? Perhaps. Let's go in on it and see if we can get a nice close in focus shot. Whoa! Big fish. Another Nanomia. There's very few of the traditional, like bell-shaped jellyfish around here. They all seem to be these physonic colonies. That looks different, doesn't it? Does that have a pair of bells? Ooh, ooh. What is that? It's got no bells. Looks like a prayed tail. Oh. Are you able to suck this up, or is it too close already?
We shall attempt. Dougal, for you.
So the brachial canals are so distinct. I don't think I've seen one quite like it before. Prayed siphonophores, the bracts are also quite distinctive between the species. So, well, that's good. even without the swimming girls. There we go. Nice.
Got it.
Yes. Okay. Should be able to tell the species on that. Excellent. So they're two competing species occurring in the same water mass. Two different taxa. Oh, that was a rhizarian, maybe a radiolarian with a spiny glass skeleton. Definitely a lot of action happening here. Lots of life in the midwater today. Mostly siphonophores. I don't think we've seen a single pseudophore yet.
So Dougal, in terms of our track, we've started off, we've headed initially in a southeast direction. Now we're sort of trending back up to the northwest currently, just to make sure we don't get too far from our first waypoint for the benthic dive. But we're doing well time-wise, 245 metres.
So based on the other dives we've done, you'd be expecting the bell-shaped Ctenophore, Thalassocalis, to be turning up soon. They're very transparent. We've been kind of moving quickly because we've been trying to quantify the number of Nenomia colonies that are here, but if we go too fast, we might not be able to find and stop on the Thalassocalis if they're around. So yeah, this is a pretty good speed, I think. Probably the krill will be a little bit deeper than this. But that seemed to be a very tight layer of Nenomias. Possibly a patch. We really don't know what the three-dimensional structure. It's an oikofluid lavation house. The three-dimensional structure of plankton communities is like. We do know that they're not distributed evenly in the water, which means when we try and quantify animals using plankton nets, and we're possibly dragging the net through 500 meters of horizontal space per layer at a minimum, you're integrating the numbers of animals and only getting an average value. Using the ROV like this, we can say, okay, at the same depth, there were places where the plankton was at X amount of density, and other places at the same depth where it was very different. So we can get more of an idea of the range of animals per cubic liter or cubic meter that are occurring there. And because the midwater is a mishmash of all these different patches of different planktonic organisms and particles, you get things happening like, say, the sperm whales off Ogasawara in Japan in a completely oligotrophic area. There are sperm whales and giant squid that congregate there. If you look at the amounts of plankton that are there as we calculate from plankton net tows, there's not enough food to support them. But when you go down with something like an ROV, where we can see in patches, there's actually like huge numbers of animals just concentrated in small volumes. And if the animals are foraging within those volumes, then suddenly the calculations as to how much energy they expend searching for food and what they're actually gaining when they eat an animal makes a lot more sense. Another Nenomia here. Nenomia have been found to be a important prey item in sunfish, mola diets by attaching video cameras to the heads or sides of sunfish and watching them feed in situ. And they're chomping up on things like this Nenomia as they're looking for larger prey. The great thing about eating a siphonophore is if you do eat one, you're also eating all of those stomachs, many of which will include prey as well.
So Dougal, we're just going to accelerate a bit for a moment just to put a bit of distance between us and the ship. I think the current was pushing us. If it's not an issue, just a quick relocation, then we'll slow down again.
I shall close my eyes.
That's one. Don't get dizzy. Yeah, we're not dropping, we're maintaining the same depth, so we don't miss anything.
Roger that. Take us into hyperspace, Chewie.
That's the one.
We've got James joining us from Japan too. Welcome aboard, James.
He's here looking for the first midwater zoanthid. So definitely we're below the layer of all those nanomias. Even at this speed with those numbers, and now that we have a grip on their morphology, we'd be seeing them floating past.
Okay, resume. Sorry, just resuming normal transmission.
Collecting with a plankton net, say you were sampling the 400 to 200 meter layer in a single net, the numbers of animals, the nanomias that you'd be counting would be far less when you think about how thin a layer that was and the numbers that were in there. You really get an idea of how useful these ROV dives are to correctly characterizing the midwater. What we really need is a camera system that can visualize the small particles as well, the foods such as copepods and krill, concurrent with the video camera, which we were planning to bring on the current mission, except that due to COVID related problems, I was unable to join the cruise, and only some of my equipment was able to be brought with us. We had been going to deploy a shadowgraph camera system that would image large volumes of water, but be able to visualize small organisms concurrently with the regular video camera transects on the dives. Unfortunately, that was not possible. So we'll be looking at the 4K high resolution video and trying to see what the animals are based probably mostly on behavior. Copepods tend to jump many body lengths whenever they escape. Krill tend to donut or twirl. Amphipods swim at a reasonably stable pace, just zigzagging through the water as they go. So even if the resolution of the images is not so great, we can still tell what the major groups are by their behavior.
Dougal, you mentioned this shadow camera that didn't make it on board. Can you just explain that a bit?
So the shadow camera, it is two pressure housings with sapphire glass ports or windows on them. There's an LED, a blue light LED in one, and a camera in the other. And they have a collimating lens in front of the glass of each. And what this does is it takes the pinpoint source of the single LED as the light source, and it makes all the light that comes out of the housing be completely parallel. So not radiating out like normal light would be like this, but instead every light ray is parallel. And then that's taken in by the collimating lens on the camera housing to get images of the silhouettes. Oh, this is a big prayed Siphonophore, it looks like. Can you take a look at the neck nectosome, yes, that's nice. See if we can see. It looks like it has two globular extensions into the mesoglia of the somatocyst. Yes, this is Desmophis annectans. Desmophis annectans. Desmophys annectans. Very nice. So some of the animals we can tell because they have macromorphology that lends itself to in situ identifications, such as this. It's a prayed siphonophore. It has two opposed swimming bells. That sphere of white material you see inside each of the swimming bells, this is where it stores its food or energy. And it supplies that through this very long tube, like an aorta, an artery that goes right through the animal to all of these chlamydia. Each of those white spots is a stomach. So you're looking at over 100 stomachs attached to this siphonophore colony, each one of them with a tentacle, which it dangles down into the water to collect or prey on small crustaceans, probably. As we look along the cyphosome, we can see how many of those stomachs actually have prey items in them. See whether there are any that are shaped like shrimp or shaped like copepods. I can't see any that are bulging in a very different way to the ones next to them. So I'd say it's mostly, most of the stomachs are probably empty. It's certainly swimming, trying to get away from us. Very nice. So all of these images we're collecting here are going to be put into a machine learning database so that in subsequent surveys, people will be able to ask the artificial intelligence, so what's this? What's that? If they're not experts at identification using images themselves. We're also, for the animals we're able to sample, we'll be sequencing them to try and get DNA barcodes that can tell which species is which based on a short DNA sequence. And by combining these images and DNA sequences, make a toolkit for people who are not specialists in midwater jellyfish, so that they can take the little pieces of jelly that come up in their plankton nets, and they will still be able to identify the species based on the DNA barcode. Or if they send down drop cameras or other instruments to visually record the animals there, they'll be able to, ooh, wow. That's a Physonex cyphosome. I couldn't tell if it had a nectosome on there as well. Possibly we'll see that. Oh.
It's like little red, maybe squid or shrimp. It's leaving little trails, little smoke trails in the water.
Oh, really? It's squid ink.
Yep. It's definitely a squid of some kind.
Oh, yes, it's popping off pseudomorphs. Yes. So each of the ink ejector it's making are roughly the same length and thickness as the squid itself. So it's leaving decoys as a Forscalia, Formosa, it looks like. Leaving decoys of itself as it escapes. Sometimes they squirt when they run out of ink, and then they double back and hide in underneath the ink.
Little octopus.
This guy's looking at us. Its arms raised, keeping them out of the way of its eyesight. It's staring at us there with binocular vision.
I think it's an octopus, Dougal.
Oh, really?
Yeah, we're getting closer in.
Yeah, my video is very.
No, we're saying squid here.
Back to squid now.
Yeah, back to squid. It was just the orientation. So you can see it's flapping, its flaps going crazy, like a little Dumbo.
And it's got its mouth pointing to us. Oh, there was a Medusa.
That's tiny.
Body and legs are all flayed out to try and decrease the speed at which it's sinking through the water. Larger water resistance as it sinks.
Squid. Yep.
Reddish body.
There we go. Bit of light on that now. Look at that.
Oh, wow.
It left a lot of ink.
I think he'd
trails behind.
Lost it all.
Doesn't seem to be covered in bioluminescent organs.
1 centimetre maybe. If that. Yep.
Does it have like little spots all over the mantle?
Yeah, it looks to be little. I'd even go so far as little nodules.
I wonder if the eyes are different sizes.
Right. Okay. Interesting orientation.
Certainly not a species I've seen before.
Okay, it's a new one.
Be a southern hemisphere species.
We're just reorienting the ROV to see if we can see the other side.
Roger. It's got the same posture that some of the what do they call them? Strawberry squid have. Cockeyed squid. And if we can see if one of the eyes is large and the other is small, that should allow us to see if it's a cockeyed squid or not.
So this is a view from the rear. You can definitely see those bumps on its mantle.
Man, I think this is great from where you're sitting.
Yeah. Okay. He's rotating with us. Doesn't want to show he's on the side.
If it is a cockeyed squid, like one eye is evolved to look at some things and the other eye other things. And obviously the one that lets it see the ROV, the bright light best, is the one that it's pointing towards us and making sure it always keeps us in sight. So we may not have much luck. Possibly a cockeyed squid. We'll have a look at the high quality video later on, see if we can ID that animal. Certainly some interesting behavior. Story about cockeyed squid. They used to think that these animals orient in mid-water with the large eye pointing down and the small eye pointing up. The large eye being large to gather light and see the point sources of bioluminescence below it, and the small eye looking upwards to see the downwelling light. Smaller because the light is brighter from above. But this was based on an animal caught in a plankton net. And we found through in situ observations that actually there's a whole slew of different ways they orient, including being vertical, the body being vertical with the arms pointing down, and that the animal caught in the plankton net was pretty sick, about to die. And the reason the large eye was pointing downwards was because it was heavier than the small eye. And so it was kind of just lying there, feebly beating its fins and almost dead. And we thought that was the way they oriented in the mid-water. So it just shows how important it is to actually get video of their in situ behaviors to infer what the correct ecology is. So we're down here at 320 meters depth. There's another Physonex Siphonophore. Looks like a Galma okini, perhaps. It has a very thick cytosome, the same width as the nectosome. See whether it's orangish if we get closer. The float is up to the top of the nectosome. Looks like it's pulled it in between the upper nectophores. And the bracts on the cytosome, that bottom half of the animal with all the bright white stomachs on it, they're interlocked and protecting the stomachs and the gonads, the sexual units from us. Thinks we're going to eat it. Okay, thank you. I'd be expecting some krill turning up round about now. Perhaps, and I still haven't seen a Siphonophore. Although my video is pretty blocky, so I might not recognize one even if I did. We'd really have to stop.
Hey Dougal, we've seen quite a few arrowworms or Ketignates. Can you tell us a bit about those?
Ketignates are voracious predators. We still don't know exactly where they fit in the higher order of life because they're genetically they're a real mishmash, fast evolutionary rates. They're transparent animals. They have opalized jaw, usually curved talons on either side of the mouth. They have little eyes to see what they're about to eat. And they eat pretty much everything that moves. And they also eat carnivory. Cannibalistic behavior is also known for Ketignates. So I was diving in the manned submersible Shinkai 2000 once, and I was looking at all these L-shaped objects in the water column, and I couldn't tell what they were. And when one of them came close enough to the window, I saw that it was a Ketignate that had chomped into another Ketignate and was holding it in its jaws just behind its head, like it had it on a neck hold, a stranglehold. And they were all over the place, these Ketignates feeding on each other. I guess there wasn't enough food for them to be eating copepods at that time. But very, very voracious animals. They normally lie in wait, floating with their gelatinous bodies. And when they sense something close, they just latch on, speeding forward in the water quite quickly. Shooting like an arrow, which is where the name arrowworm comes from. Like a fluid house. Ketignate means basically hairy jaw. And it's because of those curved spines that look a bit like hairs coming out of the mouth, on either side of the mouth. "340 meters depth. Looks like the altimeter is showing up now, so the seafloor is 100 and a bit meters below us. You can see some oikopleurids. I'll have to be dependent on you guys to find animals to stop on, because I've got jumpy video.
Roger that, Dougal. We'll keep an eagle eye out.
No dinner plate jellies or misses yet, I don't think. I haven't seen any Siphonophores. So far during this dive, there's been Siphonophores all over the place. floating and swimming colonies of jellyfish polyps, each with their own specialized function. They all develop from the same egg, same fertilized egg, same larva. And as they differentiate, kind of like the organs in your body, like someone becoming a liver, someone becoming a stomach, someone becoming a brain, except in the case of siphonophores, they can live for quite a long time without any of the other organs there. So it'd be like your stomach could go on living all happily by itself for many days, as long as it was catching food. So it's a higher level of independence between each zooid than we have in the human body, but they do all depend on each other to function as an organism. You need to have one of the gastrozoids to ingest food and digest it. You need to have the swimming gills to keep you mobile and floating in the water. You need the reproductive gonozoids in order to reproduce. Looks like there are ctenophores around. Colleague in Japan has just piped in saying there was a lobate ctenophore passing by. Oh, what's that? Was I don't know, a fluid or a ctenophore? Looks like we're stopping on something. Looks like an fluid house perhaps again. That's an oikofluid house. You can see the bilobed inner filter structure and the external outer filter structure. Is the animal in? Can't. Possibly that bright white spot in the center in between the two lobes is probably the animal, and it's got a transparent tail which is pointing down towards us there. That allows us to get the genus, that's an Oikopleura of the larvation, based on that bilobed inner filter, which is smaller in size than the outer filter, but not hugely different in size, as would be the case with Bathycordius. And these animals, they produce these houses. The outer filter is to keep all the large particles out from clogging the inner filter. The inner filter has a mesh size that filters out particles that are small enough to fit in the mouth of the larvation. And it keeps an extra feeding filter deflated underneath its chin. And when the outer filter of its feeding filter gets clogged, it'll discard it, which means it swims out of the house and lets it sink by itself down to the seafloor and the layers below, and it inflates the house stored under its chin in a couple of flicks of its tail. That's another Oikopleura, and starts filtering water again very happily. They can eat very small particles, all the way down to cyanobacteria in size, which means they're very efficient at gathering food even when there's not a lot of food around. This dive we're doing now, there seems to be quite a lot of food around. There are many siphonophores in the upper layers. So the water column is not dominated by tunicates, which would normally be the case if there's not so much food around. Possibly the tunicates have even been eaten by the large numbers of siphonophores that we've been seeing. 375 meters depth, 80 meters above the seafloor. I would imagine we'll be having krill, possibly visible by now. You should be able to tell a krill from another marine snow particle by the way they swim in donuts, spinning around and around on their axis. with their back pointing towards the center of the circle as they spin around. I take it the number of siphonophores floating around is a lot lower than it was 100 or so, 150 meters shallower than here, although that looks like it might be one.
So we're only about 70 metres off the seafloor now. You're right, we're not so many siphonophores now, but still plenty of life out there.
Okay. My comments will be a bit less now because I can't actually make out very much. Could the YouTube video right now actually be higher bandwidth? Should I switch to that?
You could try that. We're running both.
That should be an extra fluid on a fastener. That's an extra fluid house. Is that a fastener? Which ROV dive number are we on right now?
392. 392. Here's something different right now, Dougal. Is this...
Ah.
Do you have vision?
Yes. Looks like it might be Siphonophore.
We'll just try and steady on and...
Oh no, it's an oyster fluid. We're looking at it from like down
yeah.
through the animal right on the top.
Okay.
I'm not sure.
Yeah.
The video's not quite up to it.
So we're now 70 metres above the seabed. Dougal, how would you like us to run from here? Happy to keep drifting at this depth or gradually drop, or actually have a look at this. Seem to have lost. No, I lost audio from Dougal. Sorry, Dougal, we've lost your audio in here. Might do a chat.
Yeah, so,
oh, it's better.
That was a lamport case, possibly. We can tell it's a lobate Siphonophore or cone jelly. We'll be able to tell what it was by looking at the tentacle bulbs, if they're on either side of the mouth or up at the top of the stomach. It could have been a Thalassocaris as well, which is a bowl-shaped or cup-shaped Siphonophore. Should be able to tell that by looking at the high-quality video later on.
That's great.
Okay, so we're 60 to 65 meters off the bottom. Slowly heading down. Big target there. Lake Victoria in the foreground. Is that a salt chain in the distance there? Looks like it, yeah, looks like a chain of salts. So these look like Siphonophores when you're looking from a distance, because it looks like each of those stomachs, and there are stomachs on the salps as well, those bright white spots. The difference is there's no tentacles hanging down. Although they're gelatinous, they look like jellyfish. They're actually more closely related to fish. Although they don't have a backbone, they do have a notochord. So like what equates to a spinal nerve running along their bodies. And these are the animals that filter food out of the water as they swim. So they're eating at the same time as they swim. Good food if you're a dinner plate jelly, a solmysis, or a siphonophore. These are all clones of each other, still joined together, produced by a solitary salp. Okay. What else are we going to find? All right. I think possibly because we have other people on the Zoom channel, the video is not quite as nice as it might be. Am I better off going on to YouTube and chatting in there, or how should we do this? I'll just keep my microphone on and look at the YouTube feed. I'm not muted now, am I? No, okay. Lobate Siphonophore. Looks like a euhamphaid. See if we can see if it has any filaments coming out of the right-hand end of the animal there. Looks like there's a krill. Oh, a tomopterid worm just above it, coming in front. And either a Keohimea or a euhamphaea. If it has filaments, it's a euhamphaea. Without, it's a Keohimea. A nice big flattened lobate Siphonophore. Siphonophores, rather than having the stinging cells or nidae, nematocysts that cnidarians have to catch their prey using venom, Siphonophores have what are called colloblasts, which are like ping pong balls on a string, where the ping pong ball is covered with flypaper. There's lots of krill around in the water there, many, many krill. And krill are good food for euhamphaid Siphonophores. Seen them in the stomach in multiple occasions. So the comb jelly is where the food is, as would be expected. Krill. copepod. ROV seems to be going backwards slightly now. Are we repositioning to the seafloor or having a pilot change, perhaps?
Hey Dougal, Steph here. Can you hear me?
Dougal, we've just had a crew change over here. We've got Chris and Adam as our ROV pilots.
(388 meters depth). That's another oikoplurid house. Looks like another oikoplurid. There we go. That's nice. Particles are coming into the camera. A lot of particles around. What do we have here? Clear, transparent, and white